Module 2, Toxicology. 1. Introduction to Toxicology. Toxicology is the science of poisons, but it's much broader than just identifying harmful substances. It is the study of the adverse effect of chemicals, physical agents, and biological substances on living organisms, including their nature, mechanisms, detection, treatment, and prevention. In other words, toxicology answers questions like: what makes a substance toxic? How much exposure is too much? How does a toxin cause harm in the body? How can the effects be prevented or treated? One of the central principles in toxicology, famously stated by Paracelsus in the 16th century, is: the dose makes the poison. Even water can be toxic in excessive amounts, while extremely toxic substances like botulinum toxin can be used safely in controlled, tiny doses. 2. Scope of toxicology. Toxicology spans many fields, and often overlaps with pharmacology, chemistry, medicine, environmental science, and public health. Major branches include: descriptive toxicology, observes and describes the toxic effect of substances, often through animal testing. Mechanistic toxicology, studies how toxins produce their effect at the molecular, cellular, and biochemical levels. Regulatory toxicology, focuses on setting safe exposure limits and enforcing safety laws. Forensic toxicology, applies toxicology in legal context, such as detecting poisons in criminal investigations. Environmental toxicology, examines how pollutants affect humans, animals, and ecosystems. Clinical toxicology, deals with diagnosing and treating poisoning in humans. 3. Types of toxic agents. Toxic agents can be classified in various ways. 3.1 By origin. Chemical toxins, metals, lead, mercury, solvents, pesticides, industrial chemicals. Biological toxins, bacterial toxins, botulinum toxin, plant toxins, ricin, animal venoms, cobra venom. Physical agents, radiation, extreme heat or cold, mechanical trauma. 3.2 By target organ. Hepatotoxins affect the liver, e.g., aflatoxin, alcohol. Nephrotoxins affect the kidneys, e.g., cadmium, certain antibiotics. Neurotoxins affect the nervous system, e.g., tetrodotoxin, lead. Cardiotoxins affect the heart, e.g., aconitine. Pulmonotoxins affect the lungs, e.g., asbestos, ozone. 3.3 By effect. Acute toxins cause immediate effect after a single exposure. Chronic toxins cause damage over long-term exposure. Carcinogens cause cancer. Mutagens cause genetic mutations. Teratogens cause birth defects. 4. Dose-response relationship. The dose-response relationship is at the heart of toxicology. It describes how the magnitude of a toxic effect depends on the dose received. At very low doses, a toxin may have no noticeable effect. As the dose increases, effects become more pronounced. At high enough doses, little effects occur. 2 important concepts. Threshold dose, the lowest dose at which effects are observed. LD50, lethal dose 50, the dose that kills 50% of a test population, a standard measure of acute toxicity. 5. Factors affecting toxicity. The severity of toxic effects depends on several interacting factors. 5.1 Dose. As mentioned, dose is the single most important factor. Small doses might be harmless or even beneficial, while large doses are dangerous. 5.2 Duration and frequency of exposure. Acute exposure, single or short-term exposure. Subchronic exposure, repeated exposure over weeks or months. Chronic exposure, continuous or repeated exposure over years. 5.3 Route of exposure. The way a toxin enters the body affects its potency. Inhalation, gases, aerosols, fumes, often rapid absorption via the lungs. Ingestion, food, drink, passes through the digestive system, may be metabolized in the liver before reaching the bloodstream. Dermal contact, skin absorption, depending on the chemical's properties. Injection, direct entry into the bloodstream or tissues. 5.4 Individual susceptibility. Factors like age, sex, genetics, nutrition, pre-existing health conditions, and lifestyle can influence how a person responds to toxins. 6. Absorption, distribution, metabolism, and excretion, ADME. Understanding ADME is key to knowing how toxins behave inside the body. 6.1 Absorption. Inhalation, rapid due to the large surface area of alveoli. Ingestion, mostly in the small intestine. Dermal, depends on lipid solubility and skin integrity. 6.2 Distribution. Once absorbed, toxins are transported via the bloodstream to various organs. Lipid-soluble toxins may accumulate in fatty tissues, others may bind to plasma proteins. 6.3 Metabolism, biotransformation. The body often modifies toxins to make them easier to excrete. This usually occurs in the liver. Phase 1 reactions, oxidation, reduction, hydrolysis, introducing or exposing a functional group. Phase 2 reactions, conjugation with molecules like glucuronic acid or sulfate to increase water solubility. Interestingly, metabolism sometimes converts a less toxic substance into a more toxic one, a process called bioactivation. 6.4 Excretion. Toxins are eliminated mainly through urine, via the kidneys, or feces, via bile. Other routes include sweat, breath, and breast milk. 7. Mechanisms of toxic action. Toxins can cause harm in many ways. Enzyme inhibition. Some toxins block enzymes, preventing critical biochemical reactions. Example: cyanide inhibits cytochrome oxidase in the electron transport chain, halting cellular respiration. Protein denaturation. High concentrations of heavy metals like mercury can unfold proteins, destroying their function. Membrane damage. Detergent-like toxins can disrupt cell membranes, causing leakage of cellular contents. DNA damage. Carcinogens like benzopyrene form adducts with DNA, leading to mutations. Oxidative stress. Some toxins generate reactive oxygen species, ROS, that damage lipids, proteins, and DNA. 8. Specific examples of toxins. Let's briefly look at a few well-known toxins and their effects. 8.1 Lead. Sources: old paint, contaminated water, industrial emissions. Effects: neurotoxicity, especially in children, anemia, kidney damage. 8.2 Mercury. Sources: fish contaminated with methylmercury, industrial waste. Effects: nervous system damage, tremors, cognitive impairment. 8.3 Aflatoxin. Source: fungal contamination of stored grains and nuts. Effects: liver damage, potent carcinogen. 8.4 Carbon monoxide. Source: incomplete combustion of fuels. Effects: binds to hemoglobin more strongly than oxygen, causing hypoxia. 8.5 Botulinum toxin. Source: Clostridium botulinum bacteria. Effects: blocks neurotransmitter release, leading to paralysis, used medically in tiny doses. 9. Toxicity testing. Before chemicals are approved for widespread use, they undergo testing to determine their safety. In vitro test, using cultured cells to study toxicity mechanisms. Animal testing, assessing acute, subchronic, and chronic effects. Epidemiological studies, observing human populations exposed to certain chemicals. 10. Prevention and treatment of poisoning. Prevention, safe storage and handling of chemicals, proper labeling, use of protective equipment, adherence to regulations. First aid for poisoning. Inhalation, move to fresh air immediately. Ingestion, do not induce vomiting unless directed by a professional. Dermal contact, rinse thoroughly with water. Medical treatment. Antidotes, specific chemicals that neutralize toxins, e.g., atropine for organophosphate poisoning. Supportive care, oxygen therapy, fluids, symptom management. Chelation therapy, binds heavy metals so they can be excreted. 11. Emerging issues in toxicology. Modern toxicology must address new challenges. Nanomaterials, tiny particles with unique properties and uncertain health effects. Endocrine disruptors, chemicals that interfere with hormone systems, e.g., BPA in plastics. Mixtures of chemicals, real-world exposures often involve combinations, complicating safety assessments. Climate change effects, changing ecosystems can alter toxin exposure patterns, e.g., harmful algal blooms. 12. The link between enzymology and toxicology. Before we close, it's worth noting that enzymes often play a dual role in toxicology. They detoxify harmful substances, e.g., liver cytochrome P450 enzymes. But sometimes, they activate harmless chemicals into toxic metabolites, bioactivation. Understanding enzymes is therefore crucial for predicting, preventing, and treating poisoning, tying modules 1 and 2 together into one continuous picture of life's chemistry and its hazards. We've explored what toxicology is and its scope, types of toxic agents and classification, dose-response principles, ADME processes for toxins, mechanisms of toxic action, examples of specific toxins, testing, prevention, and treatment. Modern challenges in toxicology.